So today I'll be talking about the effects of acute BPA exposure on skeletal muscle and adipose tissue metabolism. I will begin by talking about some background information. Bisphenols are chemical compounds characterized by two hydroxyphenyl groups. There are several types of bisphenols. However, BPA or bisphenol A is the most commonly used and most commonly researched bisphenol. They can be found in plastic bottles, in can linings, in receipts, dental sealants, and dust, to name a few, as it is used in the manufacturing of these products. However, bisphenols pose a problem since they can leach into food due to incomplete polymerization and of the monomers. So several studies have shown that BPA, for example, can have harmful effects on human health, as it's been linked to cancer, to metabolic syndrome, etc. In mice, for example, bisphenols have been shown to alter glucose metabolism in the skeletal muscle. However, whether the effects of bisphenols on muscle metabolism is due to the direct exposure of BPA on muscle, or whether there is crosstalk with other organs such as adipose tissue. So the skeletal muscle is important because it accounts for 80% of postprandial glucose disposal. Therefore, it is important in whole body glucose metabolism. The adipose tissue, in addition, is also very important for homeostasis because it's an endocrine organ and secretes adipokines, and also it stores triglycerides. So the objectives of this study was to determine the direct effects of BPA on glucose metabolism and mitochondrial function in skeletal muscle, and two, to determine the direct effects of BPA on adipokine levels in adipose tissue. So I will start with some methods. For muscle, we differentiate L6 myoblasts for 7 days, and during the last 24 hours of differentiation, we incubate with BPA at both physiological and supraphysiological levels. And in adipose tissue was derived by needle biopsy and treated with BPA for 24 hours, and adipokine gene expression was measured. So first we wanted to look at the direct effects on skeletal muscle. So what we see here is that oxygen consumption rate, which is a measure of mitochondrial function, was altered only at high levels of BPA, and in this case it was 10 to the 5 nanomolars of BPA. However, at environmentally relevant concentrations, there were no effect on mitochondrial function. Glucose metabolism was also altered and was increased with high concentrations of BPA, which is likely to compensate for lack of energy production by the mitochondria. And this again was only at high concentrations of BPA. Therefore, we wanted to look at the effects of BPA on adipose tissue, since we know that it is able to alter muscle metabolism as shown in mice models, so maybe this is through crosstalk with the adipose tissue. So by measuring adipokines, what you can see here is that adiponectin, which is really important for insulin sensitivity in the skeletal muscle, is decreased after 24-hour exposure of BPA. Fatty acid binding protein 4 is also altered at physiological levels of BPA. In addition, we looked at inflammatory markers, and what you can see is that interleukin 6 and interleukin 1 beta was also altered by BPA exposure. To summarize, we show that a 24-hour incubation with BPA is able to alter skeletal muscle metabolism only at high concentrations. However, at physiological concentrations, we see an effect in adipose tissue, which can in turn possibly affect the skeletal muscle. For the next steps of the study would be to co-culture skeletal muscle and adipose tissue. And the significance of this is to better understand the effects of bisphenols on human health, and this can help aid manufacturing regulations of consumer products. Thank you. collective knowledge, my lab in Ottawa, so the lab of Dr. Aguirre, and also Dr. Pereira's lab at Uppsala University.